Hi class, so in this video I'm going to talk to you about nerves, which is part of this section of 6.5, nerves, hormones, and homeostasis. And this video will deal with assessment statements 6.5.1 and through 6.5.6. The first assessment statement says, state that the nervous system consists of the central nervous system, CNS, and peripheral nerves, and is composed of cells called neurons that can carry rapid electrical impulses. So you got to imagine that your body is sensing stimuli and it's responding to it, and this response and this sensing system has to work very quickly. The central nervous system was shown on the title slide here, and it consists of the brain and the spinal cord. That's the CNS, the central nervous system. The peripheral nerves is the rest of the nervous system that is connected to the brain and the spinal cord, and that's shown in this image in yellow. The next assessment statement says, 6.5.2, draw and label a diagram of the structure of a motor neuron. So we'll draw the general structure of a nerve cell, which looks something like this. And this is called the cell body. And these projections are called dendrites. And they're receiving a signal either from a stimulus or from another nerve cell. And it has a long body on it, and that's called the axon. And then at the end is some kind of axon ending. That can also be called the terminal button. And signals get sent down from the dendrites into the cell body and down through the axon to the terminus, where it sends it to an effector, to something that responds to the stimulus, or to another nerve cell, so the stimulus can continue to be sent. And you can see here from the image on the left where we have all these parts labeled. Remember, there's a nucleus inside the cell, of course. It contains DNA, the instructions for the cell. And there's a myelin sheath on nerve cells to help transmit this signal down the axon faster. And your book has labeled these gaps in between the myelin sheath, and that's on page 174, and these gaps are called nodes of Ranvier. In the next assessment statement, it says, state that nerve impulses are conducted from receptors to the CNS by sensory neurons, within the CNS by relay neurons, and from the CNS to effectors by motor neurons. So this figure is showing you that you have to sense something from your environment, so in your hand, those are called sensory neurons. They are sensing the stimulus or the stimuli. That signal is being sent back to your central nervous system, which is here in this figure. And there are neurons that are relaying or transmitting that signal to your muscles or other effectors. Effectors are those systems that are responding to the stimuli and causing some change in your system. So in this case, like moving the muscle in your hand, if you notice that there's a flame here which is making your hand hot. So the relay neurons are found in your central nervous system, that is your spine. And then those relay neurons are transmitting the signal to the motor neurons, which are connected to the effector, in this case the muscle in your arm. In the next two assessment statements, it says 6.5.4, define resting potential and action potential, depolarization and repolarization, and 6.5.5, Explain how a nerve impulse passes along a non-myelinated neuron. Now, if you search for action potential animation, there's a really good animation from McGraw-Hill, which leads you through understanding how this process works with something that's moving. But I'll try to do a good job here explaining to you how you send this really fast signal along the axon of the neuron. So the resting state is, you see that the sodium potassium pumps inside the axon are maintaining a negative charge inside the axon and a positive charge outside the axon. This is done, again, as I said, through sodium potassium pumps. And the potassium ions, which are positive, are being pumped in, and the sodium ions, which are positive, are being pumped out. Now, through this pump, you're actually pumping more sodiums out than potassiums in. That's why there's a greater positive charge on the outside than there is on the inside. So you end up having a slight negative charge on the inside of the axon. When there is a great enough stimulus stimulating the cell body of the neuron, it sends a signal down into the axon, and this is called the action potential. Basically, it opens up these channels so the sodium ions want to reach equilibrium, so they flow into the axon body, causing a depolarization. Remember, the word polarity refers to a difference. So initially there is this difference in charges between the inside and the outside of the axon. But when you open up these channels inside the axon, it allows the sodium to flow in. It rushes in really quickly, and it causes an equilibrium to occur. And if that equilibrium happens quickly enough, it activates the channels that are downstream, right? The signal is going that way. So, and you can see that in this image. And this signal, this action potential, is being sent very quickly down the axon towards the end of the axon. And repolarization happens right after the action potential passes. And that happens very quickly also. The amount of time it takes for that to happen, for it to return back to its resting potential, to repolarize, to get the positive on the outside and the negative on the inside, that's called the refractory period. Now that's done by the action of these sodium potassium pumps. Once the action potential has already passed, the sodium potassium pumps pump the sodiums out and the potassiums back in. And the amount of time it takes to do that is called the refractory period. So then another signal can be sent down the axon. If it's an especially strong signal, you'll get this happening many times in response to the same stimulus. And the last assessment statement for this video is called Explain the Principles of Synaptic Transmission. At the end of the axon is this terminal button, which contains synaptic vesicles, which are waiting for a action potential to reach them so they can be released into the synapse or the synaptic space here. And you can see where we're talking about in this picture on the side. It's happening at the end of one nerve cell and at the start of another one. So if you look on page 179, you can follow the sequence here. The calcium ions diffuse into the terminal button. They reach these vesicles that contain the neurotransmitter. By exocytosis, the neurotransmitter gets released into the synaptic space. They bind to the receptor proteins in the postsynaptic cell, and they send a signal inside the postsynaptic cell, either for stimulation of that cell body or stimulation of that new dendrite. Then the neurotransmitter gets broken down, and that's not shown on this image, but you wouldn't want to keep stimulating the postsynaptic cell. So what you want to do is you want to break down the neurotransmitter into parts so it can get reabsorbed into the original axon, so it can get recreated again for the next action potential that comes along. In the next video, I'm going to talk about hormones and homeostasis. Thanks. Bye.